All right, folks, so this is 17.4, the visual physiology. There is going to be something very counterintuitive in here. I mean, it'll be normal to you after a little bit, but it's going to sound really weird and counterintuitive when we get going. So let me get started here, and this is a real blown up picture of the retina, as you can see. You can't even see the ganglion cells, but you can see the light coming in, and it bypasses these bipolar cells and hits our photoreceptor cells. Our photoreceptor cells are cones and rods. That's what they are. These are the guys that absorb light and then have a reaction to it that we'll talk about. The rods and cones have these discs, which are layers and layers of phospholipid bilayer membranes, is really what they are. So these discs right here, they're called flattened membranous discs, and the flattened membranous discs contain the visual pigments. The visual pigments in the rods is called rhodopsin, and the visual pigments in the cones is called iodopsin. But there's three different types of iodopsins, which give us our red, green, and blue uh, color perception. Well, obviously you can see all the colors of a box of crayons, or whatever you want to say about color, like the colors of the rainbow or the colors of a box of crayon. You can see them all. But we have three main types of iodopsin: the red, the green, and the blue. And it's a combination of these that allow us to see a multitude of colors. So the visual pigments, rhodopsin and iodopsin. Okay, so let's get going with what happens here. This is showing you the flattened discs. Let me go back and show you where the flattened disc is. The flattened discs are in these parts of the rods and cones. You can see them there. All right. If you've ever taken general biology or something like that, it's similar to like a thylakoid membrane of a chloroplast, where as similar in the way that it's a flattened disc or flattened membrane system containing a pigment. Like the chloroplasts have these thylakoid membranes containing chlorophyll. Well, these are flattened membranes containing rhodopsin and iodopsin. Rhodopsin for rods, iodopsin for cones. And this is a blown up picture of the flattened discs right here. And there's three of them. Obviously, there's more than three of them, but here's three of them stacked. And then the visual pigment are these little purple spots you see here. Those are the visual pigments inside the membrane. Now here's a blown up picture of the membrane. It's a phospholipid bilayer. So each one of these is made of a phospholipid bilayer. And then here's the blown up picture of the visual pigment. This happens to be rhodopsin. Now, rhodopsin is a holoprotein. All right. Well, what that means, if you recall, is that there's an apoprotein. All right. Apoprotein. And the apoprotein is called opsin. All right. And then there's a prosthetic group. Prosthetic group. And the prosthetic group is called retinal. It's a vitamin A derivative. It's the reason why you eat vitamin A. All right. So the holoprotein, rhodopsin, is part opsin, part protein, and part prosthetic group called retinal. And that's the holoprotein called rhodopsin. All right, so let's look what happens here. And this is going to be the part that's really counterintuitive. All right, so first of all, this is the rhodopsin. Your rods contain rhodopsin. Very sensitive to low levels of light. All right, you can see that its maximum absorbance is about 525 nanometers wavelength. Here are the iodopsins. These are the iodopsins. And you have the blue iodopsin, you have the green iodopsin, and the red iodopsin. Let me point out something very peculiar about these colored iodopsins. The red iodopsin actually has a maximum absorbance, not quite in the red wavelength, but in the yellow wavelength. But we still call it red, green, and blue color perception, even though its maximum absorbance is in the yellow wavelength. Yellow or orange, getting towards red. So those are the three iodopsins and the one rhodopsin. All right. Okay, this is the counterintuitive part. What happens in this, by the way, I'm going to talk about a rod, but the same thing happens in the cone. It's just that the iodopsin is the visual pigment, not rhodopsin. So I'm going to always say rod and say rhodopsin, but the same thing in the cone except for it's iodopsin. Here we go. This is the rod. The rod has a dark current, and that means that neurotransmitters are flowing. This is depolarizing, and neurotransmitters are flowing in the dark. When light hits it, it hyperpolarizes. And that's not going to sound right to you. You're going to think light hits this and it depolarizes and sends the signal. And this is what's counterintuitive. In the dark, this thing is depolarizing and releasing a neurotransmitter that is inhibitory. So the bipolar cell never fires in the dark, and therefore the ganglion cell never fires in the dark. But the rod and cones themselves have a dark current in the dark. And then when light hits them, they hyperpolarize. They stop releasing inhibitory neurotransmitter. The bipolar neuron does fire. The ganglion cell does fire. And this goes to the brain, and your brain sees the image. All right, so let's get back to this. What's going on when light hits this? Well, here's a photon of light hitting my rhodopsin. All right, when it hits the retinal, this retinal is cis retinal. That's what it's called. It's called 11 cis retinal. That's what that's called. Because it has a cis double bond in it, it's kinked or crooked right there. It's similar to why unsaturated fatty acids are kinked or crooked. It's the same reason this is kinked or crooked. It has a cis double bond in it. All right, it's called 11 cis retinal. When light hits it, it changes that double bond to all trans retinal, and it straightens out. This is really peculiar biochemically because it's one of the fastest reactions known. It only takes like a femtosecond. This only takes a femtosecond to occur. Now let me see if I can remember what a femto is. Nano is 10 to the negative 9, so it's 1/1000th of a second. Pico is 10 to the negative 12th, 1/1000000000th of a second. Femto is 10 to the negative 15th. So this reaction is 10 to the negative 15th of a second. That's how fast it is. All right, so this is a femtosecond type of reaction. Now I have 11 trans retinal right there. All right. Now what happens is this is going to change the permeability of the rod. And I want to go through the steps of why this changes the permeability of the rod. But ultimately, this 11 trans retinal is going to lead to hyperpolarization. Hyperpolarization. Details to follow. Meanwhile, I need this rhodopsin to be able to absorb another photon of light. But right now it's what we called bleached. Well, it's actually going to be bleached here, but it's almost bleached. It's not able to absorb another photon of light. So what happens? Well, the first thing that happens is all trans retinal and opsin dissociate. So here's the apoprotein, opsin. And here's the prosthetic group, all trans retinal, and it's dissociated. An enzyme acts on the all trans retinal and converts it back to 11 cis retinal, and it requires ATP. So now I have 11 cis retinal. This is the retinal I need to absorb another photon of light. But the problem is 11 cis retinal is dissociated from the opsin. No problem. I reassociate them. The opsin and 11 cis retinal reassociate, and now I have my functioning rhodopsin again. I have my holoprotein. In this area right here, we say that the rod is bleached. It can't absorb another photon of light. And this happens because light hit it. So this is what happens with your retina when bright light hits your retina and it blinds you temporarily. It bleaches all of these visual pigments, and you have to let them reform into the holoprotein before you can see again. So this is a bleaching that occurs. So here we are back to the functioning holoprotein, and you can absorb another photon of light. And the 11 cis retinal can become 11 trans retinal, and that can lead to hyperpolarization of the rods or cones. And then they dissociate, and it takes ATP and an enzyme to reconvert 11 trans to 11 cis. Then they reassociate, and the vicious cycle keeps on occurring. So really what I haven't showed you yet is what this 11 trans retinal, how it leads to hyperpolarization. That's what I haven't showed you. Here we go. All right, so this is actually not the answer. This is not the answer of the rhetorical question I just asked. This is just looking at the details of what we just talked about. Photon of light causes 11 cis to become 11 trans retinal. Remember, retinal is a vitamin A derivative. This is why your mother told you carrots were good for your eyes. You have to eat vitamin A to make this 11 cis retinal. That's why you need vitamin A in your diet. Okay? All right, now this answers your question. What happens when I have 11 trans retinal and opsin? Before they disassociate, they turn on a G protein. The G protein, do you remember what the G protein in the tongue was called? It was called gusducin. Do you remember what the G protein in the nose was called? It was called Gulf or G olfactory. Do you remember what the well, that we'll just stick with that. So the G protein in the tongue was called gusducin. Gustatory G protein, gusducin. The G protein in the nose was called Gulf. The G protein in the eye is called transducin. Transducin is nothing more than a G protein. And you've learned so many of these G protein things, it should start to become second nature. So opsin in 11 trans retinal activates transducin. Transducin activates an enzyme called PDE or phosphodiesterase. Now here's what you probably, hopefully remember. Phosphodiesterase breaks down cyclic AMP. Phosphodiesterase takes cyclic AMP, a second messenger, and converts it to AMP. It's not a second messenger when it's AMP. It's inactive. So what the transducin does is activates phosphodiesterase. This phosphodiesterase is going to break down cyclic AMP. But before it can, let me show you what cyclic AMP is doing. Cyclic AMP opened the sodium channels. This is why in the dark, this rod or cone has a dark current. It's depolarizing. It's depolarizing in the dark because in the dark, cyclic AMP is here and the sodium channels are open. But light just hit my, look what just happened. Light just hit my rhodopsin. Since light hit my rhodopsin, I have 11 trans retinal. Because I have 11 trans retinal, my transducin is activated. Because my transducin is activated, my phosphodiesterase is activated. Because my phosphodiesterase is activated, I'm breaking down cyclic, and I lied. It's actually cyclic GMP. I forgot that. Phosphodiesterase has the ability to break down both cyclic AMP and cyclic GMP. In the case of the eye, it's cyclic GMP, the eyeball. So phosphodiesterase breaks down cyclic GMP to GMP. By the way, it has the ability to break down cyclic AMP to AMP. So in the case of the retina of the eye, the phosphodiesterase is breaking down cyclic GMP. Cyclic GMP is the second messenger. All right. So now my phosphodiesterase is activated. Cyclic GMP is removed. It's broken down. This causes the closing of my sodium channels. Now I no longer depolarize, and in fact, I hyperpolarize. So in the light, my rods and cones hyperpolarize. In the dark, there's a dark current. That's what's going on here. So in the dark, there's a dark current. In the light, I'm hyperpolarized. And I'm looking for where it says it in here. As the plasma membrane hyperpolarizes, it says it right there. Now look at that. As the plasma membrane hyperpolarizes, the rate of neurotransmitter release decreases. But that's a good thing, because when this thing was depolarizing in the dark, the neurotransmitter was inhibitory. It was an inhibitory neurotransmitter. So now in the light, what I'm doing is I'm stopping the inhibitory neurotransmitter from being released, which allows my bipolar cell to fire. Bipolar fires. So the light results in my bipolar cell firing because the light hyperpolarizes my rod and cone. In the dark, my rods and cones release inhibitory neurotransmitters. And that's a really counterintuitive concept, and it's opposite of what you've been learning about neurons all along. All right, so that's the dark current, and that's how light hyperpolarizes and allows the bipolars to fire. One last thing. You might have heard in your life that pilots, a certain amount of time before they fly, are not allowed to take Viagra. Well, why the heck would that be? It's the cyclic GMP story. So you see the cyclic GMP story for the retina right here. But what happens with cyclic GMP is this. Cyclic GMP is the second messenger for a neurotransmitter called nitric oxide. Nitric oxide leads to high cyclic GMP in the erectile tissue of the penis. Cyclic GMP causes vasodilation, relaxation of the blood vessels. Blood rushes to the genitals and causes erection. All right, now what Viagra does, now the cyclic GMP is broken down by phosphodiesterase. Cyclic to GMP, PDE. What Viagra does is it inhibits that phosphodiesterase. If you inhibit that phosphodiesterase, you get increased levels of cyclic GMP, increased vasodilation, increased blood flowing to your genitals, increased erection, and everybody's happy. But this Viagra, or sildenafil is what it's called, also affects your eyesight because your retina uses cyclic GMP as a second messenger. So you can imagine if you're on a phosphodiesterase inhibitor, you no longer will break down cyclic GMP. Cyclic GMP will stay attached to its receptor. The sodium channels will stay open. Your dark current will stay firing. You will continue to release inhibitory neurotransmitters, and you won't see as well, even if a light's hitting you. And this is why pilots aren't allowed to take that drug so many hours before they fly. And this is the collage showing you the whole story. Now, that's a pretty cool story. It's counterintuitive, but you have to go through it a couple times. Replay this part of the video a couple times, because the counterintuitive part is this. In the dark, your rods and cones are depolarizing, and it's called the dark current. In the light, they hyperpolarize and quit releasing an inhibitory neurotransmitter. All right, this is just a repeat of everything I just said. That's all it is. Okay. All right, let's talk about the ganglion cells. So now, in the light, these guys hyperpolarize, which allows these bipolar cells to depolarize because there's no more inhibitory neurotransmitter, which fires the ganglion cells, and these guys depolarize. And that goes to the brain, and you see. So let's talk about these ganglion cells. Well, first of all, there's two flavors of ganglion cells. There's M and P. And M stands for magna, and P stands for parvo. And parvo means little. You've probably heard of the parvovirus. It's a little virus. You've probably heard of a parvovirus if you have a dog. Dogs sometimes get this parvovirus. I think it's called B19 or something like that. I forget. But it's a little virus. And so P stands for parvo, and it's a little ganglion cell. And M stands for magna, and it's a big ganglion cell. So there's two different categories of ganglion cells here. And just as important than that is the amount of divergence. Rods innervate, so the rod will affect many bipolar cells, which will affect many ganglion cells. You get high divergence with rods. All right, one rod will affect many bipolars, which in turn affect many ganglions. So one rod can lead to hundreds of ganglions firing, hundreds of ganglions. All right. Whereas cones, I'm presuming the yellow one's the cone. Actually, the yellow one's showing you something different. Whereas with cones, one cone typically goes to one bipolar. I can't spell bipolar, obviously. Let me spell that for you. One bipolar, which typically goes to one ganglion. There is no divergence or very little divergence. There actually is some divergence with cones. I'm just making the extreme case where there's no divergence. That means one cone causes one ganglion cell to fire. Now think about this. The ganglion cell is the one going to the brain. If there's an area in the brain that responds to one cone, that's great visual acuity. That brain is receiving information from one cone. And the cone right next to it is giving slightly different information. And the cone right next to that is giving slightly different information. So this is a tremendous amount of acuity, high acuity. Whereas with this divergence right here, this is low acuity. But it also is what allows us to see in the dark. And this is why rods help us to see in the dark. Because you have this divergence going on. So if you have 4 or 5 rods firing, that's many, many ganglion cells going to the brain. You have low acuity, but you have high sensitivity with that. All right, so the less divergence there is, the greater acuity. The more divergence there is, the less acuity there is. In addition, I have these on-center and off-center cells. If the image is hitting on this rod right here, the yellow rod, if the image is hitting my yellow rod, it's hitting my on-center cell. And my on-center cell fires, and my off-center cells don't fire. All right? But if the image is hitting my peripheral cells, then these cells fire and my on-center cell doesn't fire. And this is how I have good peripheral vision in my rods. Actually, your rods are the ones that have good peripheral vision. Your cones don't do that for you. It's because I have these on-center and off-center cells. And they differentially fire depending on where the image is hitting. All right. And this is the visual pathway. Something I want to point out, very important. That is, not all of the neurons of the optic tract decussate. Look at this red neuron right here of my left eye. The red neuron did not decussate. It went to the left brain, the left occipital lobe. The left neuron of my left eye went to the left occipital lobe. But my green neuron of my left eye did decussate, and my green neuron went to my right occipital lobe. All right? Now let's look at the right eye. The red neuron of my right eye decussated and went to my left occipital lobe. But the green neuron of my right eye didn't decussate, and that went to my right occipital lobe. So we don't have 100% decussation. But look at this. This is what's important. What's causing this non-decussating neuron to fire? What's hitting the lateral part of my left eye? The medial part of the image. That circle is non-decussating. Non-decussating, it is left brain. All right? Now, what is decussating in this red neuron? It's decussating and going to the left brain. What's decussating and going to the left brain right here, is this lateral part of the image, right here. All right? So that goes to the left brain. So the left brain is receiving this red neuron, which came from the lateral part of the image, and this, in the left eye, this left neuron, which is receiving the medial part of the image over here. So my left brain is receiving all of this, is it not? And my right brain is receiving all of this, is it not? So what I can say my left brain is receiving is this red and green. I'm sorry, red and green. I do know my colors. And what my right brain is receiving is this yellow and blue. Yellow and blue. And you can see this because of the crossing over. All right, that's the visual pathways.
All right. And then The special sense of vision involves structures of the eye, the optic nerve, and portions of the brain. As light enters the eye, it passes through the cornea. The cornea is a transparent connective tissue that covers the anterior 1/6 of the eye, like a contact lens. Its convex shape refracts the light through the pupil, an opening in the colored portion of the eye. Light next passes through the biconvex lens that focuses the light onto the retina. The retina lines the interior surface of the eye. It has two parts, the retinal pigment epithelium and the neural retina. The darkly pigmented retinal epithelium is adherent to the neural retina and prevents light scatter within the eye. The neural retina has multiple cell types that are arranged in 3 cell layers. The outer layer contains rod and cone photoreceptor cells that respond directly to light. In general, cones are used for high spatial resolution and color vision. In contrast, rods are used in poor lighting conditions and have low spatial resolution. Rods and cones both translate light energy first into a biochemical message and then into an electrical signal that is passed to cells in the inner nuclear layer. The cells of the inner nuclear layer further process the signal and then transmit it to the ganglionic layer. Axons of the retinal ganglion cells, of which there are approximately 1 million, course along the inner margin of the retina and converge at the back of the eye to form the optic nerve. The optic nerves attach to the ventral aspect of the brain at the optic chiasm. From here, the optic tracts extend around the brainstem to the lateral geniculate nucleus of the thalamus. Most ganglion cell axons from the retina end at the synapses in the lateral geniculate nucleus. Visual information leaves this area of the thalamus in the optic radiations. Axons in these radiations end in the primary visual cortex of the cerebral occipital lobe. Here, visual information is perceived as an image.
All right, so that's a wrap up of the eye.